Vanakkam. Welcome to this video on classics in neuroscience. We have been looking at signaling molecules or the chemical signals, the first neurotransmitters in the brain. In this video, we will look at what were the early evidence that was presented to suggest acetylcholine as an excitatory neurotransmitter, and the early evidence that was presented to support the idea that glutamate was an excitatory neurotransmitter. The first evidence to suggest acetylcholine was an excitatory neurotransmitter in neurons, between neurons, came from the Renshaw pathway. Let me very briefly describe this. Let us say this is a motor neuron, an alpha motor neuron that supplies the muscles. For example, I am going to, this is one part of the axon, and let us say that is the muscle. This is the alpha motor neuron. Let us say that one branch of this alpha motor neuron synapses with another neuron that I am going to now call as the Renshaw cell. Okay, whose dendrites are receiving information from the alpha motor neuron, and this Renshaw cell inhibits the alpha motor neuron that excites it. So, the alpha motor neuron causes an excitation of the Renshaw cell, and the Renshaw cell inhibits the alpha motor neuron that excited it. Actually, the Renshaw cell does more than that. The Renshaw cell inhibits the alpha motor neuron and the alpha motor neuronal pool that causes similar mechanical action. I will give a more detailed video from one of my previous courses that will give you a picture of how this works. This is the brief physiology of what happens at the Renshaw cell. So, remember one thing, remember Dale's law? Dale's law suggested that if I can show that in an axon the neurotransmitter is acetylcholine and it is excitatory, it is going to have a similar action in other branches of the same axon. So, here acetylcholine is present, here also acetylcholine is present, but remember the response depends on the receptors of course, but presynaptic unity requires, as per Dale's law, presynaptic unity requires that acetylcholine is released from all the axon terminals, whether that may be with the neuromuscular synapse as it is happening in the red connections that we show here, or whether it is happening at the Renshaw cell. Regardless, the chemical that is released is acetylcholine, and acetylcholine acts as an excitatory neurotransmitter from the alpha motor neuron to the Renshaw interneurons, and these interneurons provide inhibition to those alpha motor neurons that excite them. Okay. This is similar. So, this is in alignment with the expectation of Dale's law, and this is as one would expect, the function is as one would expect. So, this suggested acetylcholine is an excitatory neurotransmitter. So, people started searching for other examples of acetylcholine as an excitatory neurotransmitter in other places in the central nervous system, but that was not easy to find. So, the search continued for other chemicals other than acetylcholine. So, now we look at some evidence for suggesting glutamate as an excitatory neurotransmitter. Until 1950s, practically there was no evidence or very limited evidence suggesting glutamate as an excitatory neurotransmitter. Some people, Curtis, however it is pronounced, suggested or found that injection of acidic amino acids like aspartic, glutamic and cystic acids by a technique called as microiontophoresis caused repetitive neuronal discharges very similar to the effect of acetylcholine on Renshaw cells. However, John Eccles was very vociferous in accepting this hypothesis, because John Eccles said that this is not specific, that you are sending a whole bunch of things and something is happening, you are observing the something that happened as a result, and then you are saying this is because of some action of some of these things. You are not providing a specific one-to-one correspondence, because that is what is required or demanded by the criteria suggested by Eccles and Dale's law, right. So, Eccles in his report or in his response mentioned the following. I am going to read from the book. He noted that the authors argue against identification with the synaptic transmitting agent and suggest instead that they are non-specific excitants that act at receptor sites on subsynaptic membrane other than the site of action of synaptic transmitters. This suggests how cautious people were. The early investigators were very cautious and very stringent in how they accepted arguments or how they accepted evidence. So, they were very particular about not accepting this set of arguments because it contained a whole bunch of acidic amino acids like aspartic acid, glutamic acid, citric acid, and a whole bunch of things, and there was some response that was found. So what? This was the question that was posted by Eccles to this research group. So later Curtis and Watkins, in follow up to their original studies, suggested that in the cat cerebral cortex these amino acids acted as powerful excitants. That they suggested that NMDA, what is NMDA? NMDA is N-methyl D-aspartic acid, is it not? Acid was some 10 times more powerful than glutamic acid in exciting the membrane. But somehow this idea faded from the memory. Somehow this idea was not receiving more favor in terms of experimental evidence, and it was later found that indeed glutamate was the dominant excitatory neurotransmitter in the brain. The main evidence to suggest glutamate or acidic amino acids activate receptor sites came from the work on crustacean muscle fibers. I think this is work by Takeuchi, I think 1963, I think Takeuchi 1963, that suggested that local application of these acidic amino acids caused excitation of the receptor sites on the crustacean muscle fibers. Indeed, some other studies from 1959 that showed that muscle contractions induced by brain extract that contained these amino acids, these acidic amino acids, were seen. This study I think is by Van Hereveld and Mendelssohn, is that not? This is citation of this study. This is 1959. In 1972 it was cited by the study. Only much later, towards the 1980 or the 1980s or so, is when glutamate was acknowledged or recognized as the dominant or the primary major neurotransmitter, excitatory neurotransmitter in the brain. What was the reason why this was not accepted earlier? Because the evidence to suggest this was piecemeal, arrived in discrete packages, and it was not conclusive. All the criteria were not met by one experiment, different criteria were suggested by different experiments. So there was a whole bunch of skepticism in accepting whatever evidence that was presented, because these came from different corners of the world at different points in time, satisfying different one of the criteria. Because of this reason, there was considerable amount of resistance to accepting glutamate as an excitatory neurotransmitter until the 1980s, when it was conclusively shown that glutamate was indeed an excitatory neurotransmitter, was indeed the major excitatory neurotransmitter of the brain. So, with this we come to the end of this video. In this video we looked at some early evidence that was provided to suggest acetylcholine as an excitatory neurotransmitter in the central nervous system, and evidence for glutamate as an excitatory neurotransmitter in the central nervous system. Thank you very much for your attention.